everybody. I'm Gene Hyde, Head of Special Collections, and with my colleague Ashley Whittle, and we welcome you to the Ramsey Library Brown Bag Talks. Today, we're excited to have Camila Figueres here talking about her research. Camila?
Thank you. My name is Camila Figueres. I am a new diversity fellow at the biology department here at UNC Asheville. I'm very excited to start my work here and be involved with insects, plants, and nematodes. So today I'm going to talk about insects, plants, nematodes. Oh my, how they interact, how they communicate. So plants talk. And better than that, when the plants talk, other organisms could hear, listen, everything, and do some behavior that we can check and use this in our favor to help the plants be successful, especially in an agriculture situation. So what I'm going to be doing here today, I'm going to give you some background about this 3 special things that I like to work with. And after that, show some examples from my research that used to study these interactions in different crop systems and with different insects and different nematodes. Thank you for the opportunity. So first, about my background, I was born in Brazil. I grew up there, did my undergrad there. And after I did my master degree in the Amazon forest. And after that, I had the opportunity to go for the first time here in US when I start working more specific with plant physiology and understand what's happening inside the plants and how they are so smart and can be able to defend themselves without moving or running. So it was very interesting. And after that, I went back to Brazil and start my PhD there with some knowledge, a lot of knowledge about insects, and very curious special about soil situation and how organisms could communicate in this specific environment, which is very hard to see or move around. So during my PhD, I started my PhD in Minas Gerais state in Brazil, and I went to Florida. I received a scholarship to go to Florida and do some research there. After that, when I finished my PhD, I had the opportunity to go to Cornell and be a research there, faculty research there for 3 years before I moved to here, where I am now. So I'm gonna go with more details about each place, what I did, and using this as an example. So first, let's understand this beautiful communication. We have this plant here. On this case, it's a drawing from a corn plant, and some insects who is beneficial for the plants, other insects that is a problem from the plants, and above ground and below ground the plants. I'm going to be using a lot those terms here, so I would like to be clear about this. And what I'm going to be doing, I'm going to be talking how the plants could communicate on this environment and how this beautiful insects, those nematodes and other organisms could recognize this communication and use this to be around the plants. So first, to understand the system, we have the plant, correct? And the plant can move, can talk, can run. So they communicate somehow. And the way that they do this most of the time is using chemical signs. And those chemical signs and other defense, but on my case, when I'm working with the interactions, I'm very curious about the communication by volatiles that the plants release in different situations. So when plants are growing up or start having some competition or start having some damage by herbivory, these plants try to defend themselves to release some toxins or sometimes start defense or leave parts of the plants that it's going to go out so this plant can have more energy in other parts and be able to survive on that situation. Insects. Insects, they are very interesting because different insects have different abilities. And when we study interactions, we need to know exactly what kind of insects we are working with and how they feed, how they communicate with each other, if they are social or if they are cannibalist. So all depends the system. Most of the insects that I'm going to show and talk today, those are insects that damage the plants. Those are herbivorous. And most of the times they damage the plants above ground and below ground, just because I'm interested to understand if the effect that we have above ground could influence below ground response. This example here, it's one of the projects that I'm working for more than 3 years now, and I'm still working here at UNCA. It's a chestnut weevil. They are an important pest in chestnut plants, American chestnuts. And here we have the female with a huge buccal and the males here. This is a juvenile stage. And they damage the nuts. I'm going to go with more details on this specific one. So the other organism that I'm going to be talking here are nematodes, okay? Nematodes, we can have nematodes who affect humans, correct? We have a lot of damage worms for us. And we have nematodes that can be a problem for plants. And we have nematodes that is beneficial for the plants too. So the nematodes that I'm going to be talking here today are those beautiful ones here on this video that they help the plant to kill insects. Those nematodes, what they do when they found a very healthy insect, they go inside those insects. When they are inside those insects, they release a special bacteria, a symbiotic bacteria, and this bacteria kills the insect, not the nematodes. And when the insect is dead, the nematodes start reproducing inside this dead insect and produce more and more and more. When the body of the insects are full and they don't have any more resource to feed inside the insects, they start going out again and trying to find a new host, okay? All these things happen on the soil. They are very sensitive with lights and moisture, so they need to be on the soil. So the way that I decide to work with this, I decide to study plant defense pathways and below ground interactions. So I decide to combine all the knowledge and all the things that I love to study, that it's insects, microorganisms, and plants, and do some cool research to understand this system. This is first example here is a corn plant with different organisms interacting with the plant. And here it's a citrus plant system that I'm going to talk more about it. Those are the two systems that I worked during my PhD. So when I started working with this during my PhD, I was interested to understand how plant defense pathways can influence the plant environment interactions with different organisms and not specific above ground or below ground. I want to know the interactions, how if I have some stimulation on the plant above the soil, I can have some response below the soil. How this happened, this is possible, this situation happened, was my big question, my big first question. So for this, my first question for my thesis was, can plant pathway stimulation above ground influence the response below ground? Until this point, we didn't know. We know that when we stimulate, we have some response, but not the interactions above and below ground. So it was the first system that we decided to study. I decided to work with this insect here. It's a beetle, Diabrotica speciosa. It's a big pest from different crops in Brazil, special for corn. And I use corn plants and entomopathogenic nematodes that it's a very good biological control for this specific pest. So on the system, I have the beautiful plant here, corn plant, and this insect, it's a pest above ground, like I said. So the adults damage the leaves here, and the larvae, it's a big problem in roots. So in corn situation, if you have roots damage, you're going to have a big problem because all the nutrition comes from the roots, and this part that keeps the plant stable is the roots too. So if you have a problem on the roots, it will be a big problem for the plant. And we add the entomopathogenic nematodes on to control this, okay? The results that we have, we figure that when the plants are damaged by the Diabrotica speciosa above and below ground, this plant starts to signalize a specific thing that the nematodes are able to recognize and go to the plant to help the plant to control this pest. So it was very interesting. I figured out, I answered my question, but I decided to, what's going on now? What's going on with the plant? How the plant can do this? What's going on inside the plant that we have this response? So for this, I went to Florida to the part of my project there, and I decided to change the system. So corn crops or annual crop, correct? It's fast growing, fast development. And I decided to try with a perennial crop. So I went Florida. Citrus is huge in Florida. So why not start trying this in citrus? So my first test was trying to figure out if we can stimulate above ground plant citrus. I have some response below ground the same way that I had with corn. And yes, I had the same stimulation when have some problems going on with herbivory problems going on above ground. We have, yes, response below ground. But what kind of response we have? What plants are releasing that the nematodes are able to recognize and come to help. So to do this experiment, we sprayed methyl salicylate. It's a specific chemical that stimulates the plant defense pathways, okay? And at the same time, other treatments, we use the insect herbivory too, above and below ground in plant citrus, and we add the entomopathogenic nematodes and see if those nematodes will respond for this chemical. And our big goal with this part is first figure out if the citrus plants will respond the same way, and the second part, figure out why and how what's a specific thing that the nematodes are answered. And we discovered that when we stimulate above ground by methyl salicylate or by herbivory, we have a below ground response. In addition with this, we could figure out what kind of volatiles that is related with this. On this specific case, was the D-limonene responsible for the response? And we had several tests after that to confirm if was the limonene that was responsible for attract the nematodes. And the way that we did that, we did a bunch of volatiles collection samples from the plants. I'm gonna show some examples now that we used to do this. We're still using the same methodology now, just improve a little bit the methodology, but still the same way. So what NemaLab is doing right now, okay? NemaLab, it's the lab that I am in charge right now. We have two big areas that we like to study, involved interactions between plant, microbes, and insects. First is understand below ground insect interactions, more basic research trying to understand the system. And the other part is using this knowledge as a biological control for agriculture pests. So have a way that we can apply this on the field and use less chemicals. So one example, for example, that we use to study basic research, we are doing some experiments when we are trying to understand the communication between nematodes, because if we know how to manipulate the communication with nematodes, why not improve this and keep those nematodes near the plant? So before the pests start to be aggressive on the plants, we already have the nematodes around the plants. To do this, we need to figure out how they connect with each other. One experiment that we are doing here, we have a line here. When we add a specific chemicals, we call those pheromones that we can use as aggregation pheromone or repellent pheromone. And we are trying to see if the nematodes respond for this, okay? And this is still going on. We are gonna have soon some results about this, but it's very cool to see the behavior and trying to use this as biological control. This is a good example from basic research that we are trying to apply this on the field. The other example, we just published this year, is with onions, and the system, it's onion maggot, the maggots in general, but most with onion maggot, that it's a huge problem for onions. They expend a lot of money with chemicals, and chemicals that is really bad for us. So we decided to try to use entomopathogenic nematodes first to control the onion maggots. We had some control, but the big thing that happens here, we could figure out that the effect of the nematodes to be near the plant was very important, not immediately, but along the generation. We had some insects that couldn't develop well. So the long-term, the no lethal effect of the EPNs was the big thing that we wasn't expect at the first moment, but was amazing to see how this could influence in long-term for the pest control. And this was very important to understand the interactions, to understand this specific effect. Yeah, this was a project that we received some grants to conduct this. The other project, phylloxera, it's a big problem for grapes. And again, it's a pest who affect above and below ground and cause disease in the plants. So now they use rootstocks to control this because don't have an effective control for this. And our plan is first, understand the insect, first hearing this insect and know how they behave, and have to understand this and understand the interaction with the plants, and maybe be using the nematodes as well as a biological control on this system. So the chestnuts, we are doing a lot of work with chestnuts on the field and in the lab when we are trying to understand the system again and see the entomopathogenic nematodes and entomopathogenic fungi. That those are fungi that we use to kill insects, specific for killing insects, and as a biological control for chestnut weevil. And at the same time, understand the environment, what's going on in different systems, chestnut systems, organic farm, conventional farm, how they do, what's the diversity that we can find on those. So the system, basically, we have above ground when the insects are, the adults are, and lay the eggs on the nuts there. And the nuts are inside the bore here. And after that, the larvae stay on the soil. So it's our goal trying to control with EPNs and EPF, entomopathogenic nematodes and entomopathogenic fungi. So to control the larvae here. At the same time, we are doing a lot of bioassays in the lab, trying to understand how they survive here. And some results that we have prove that they can stay on the soil for more than 2 years now without doing anything, just stay there until they have the good conditions to go out and reproduce. This is a project that's going on. We just submit a grant. We are waiting for the results for this grant and keep going with those experiments. This is just one example with the larvae coming out from the nuts. Here is a pupae picture that we did a trial for 2 years and had some containers on the soil, and we could see the first time the pupa stage on this insect. Those are adults again. This is the way that we are adapt to collect volatiles and understand this communication. So all the interactions, what's going on, we need some technology to understand the systems. And the way that we are doing now, we have some methodology that we can use in the lab and on the field. The same methodology could be used for both places. So we adapt, depend what size of the crop. Sometimes we need to use bags. Sometimes we use PVC pipes to collect those. We have a pump that grab the volatiles, and we have different kind of filters that collect those specific volatiles. And after we collect those volatiles, we put on those machines, and we have all the peaks that represents each kind of volatiles. And we compare this with the library, and we can know for sure what kind of volatiles are present in our samples. So here it's one way that we collect volatiles using this probe below ground, And here it's an example, here some pictures where we collect volatiles. This is a project with grass. So it was easy to put the pipe on the top, PVC pipe on the top, and combine some line here with the filters and collect the volatiles. The other cool thing that we are planning to have here soon, and we are working hard on this, is having this machine here. This machine will be very important for us to understand everything what's going on below ground on the soil. So basically what we do with this machine, we grab some soil samples in different parts or different areas that we want to study. We grab this soil sample, we add all the soil samples with some solution. Most of the time it's just water, straight water. This machine, it's a flux cytometer, and go and run all the samples like a tube, a funnel here. And inside this machine have a bunch of lasers that with different things that separate each one of the organisms from the soil in the small wells. And the cool thing with this machine, they separate the alive insects or alive nematodes, whatever you have as organisms there. The machine could separate from the soil and separate between species. So we are in the process to have the library when we are going to compare different organisms in the soil and use machine learn. This part here represents the machine learn to identify those organisms and recognize this in a real time without kill the insects, without kill any organism. Just keep going so we can reproduce those organisms and we can save the samples. So I hope the time was okay. My plan today was talking a little bit about this amazing communication that happens, have a lot of things to be done. I'm very excited to be doing some different research here at UNC Asheville, have different ideas, project ideas with the machine, understand the nematodes behavior, understand the insects in general, all the communication. And here it's an example that we use to understand in the lab how the communication below ground happens. I will be happy to answer questions about this. Thank you very much for the opportunity. Oh, sorry. I forgot to talk about my lab group. I'm very excited with this. I just started here 5 months ago, and I already have amazing students working with me. I'm missing some here. So this is from the last meeting that we had. And I have already students working on field already, collecting samples. We have Blake that he's working with flatworms and trying to hearing those organisms in our lab now. And the group is working together as a team. I'm very impressed how our students here are able to work, learn things fast, and be engaged on a cause. So I'm very proud of this lab. Now I'm done. Thank you.
Any questions?
Sure, I'll ask a question. Thanks, Camilla. That was great. A couple of things in the corn example. How do you separate the damage that's happening above ground from the damage that's happening below ground, and separate whether the volatiles that are detected in the soil are from damage above ground on the plant and those that might be on below ground?
Great question. The way that I did that, each one was a specific treatment. So I have a bunch of different combinations. I have one treatment when I use just damage above ground, another treatment just damage below ground, and always collect the volatiles below ground and check if we have something similar.
So you're able to manipulate whether there's adults or larvae feeding on the plant.
Yeah.
Okay. And one other question, do you have any idea about the distance at which nematodes can detect these signals and how far they would travel to go to a plant based on the volatiles that they're detecting?
Great question. So I did this with corn. We test in different distance, I think was 30 centimeters, 60 centimeters, different distance, and see if the volatiles will change. Was very interesting because they could recognize in long distance and was better mid to long distance than near the plants. So I think the volatiles blend was more clear for the nematodes response for long distance than short distance.
So when you talk about a blend, it's more than one molecule that they're detecting and they respond to more than one, or how does that work?
So normally around the plants, you have different volatiles going on there. And what it's interesting because have a lot of things going on there, and sometimes you don't have a good response near the plant. But when they are far away, the organisms are able to recognize this and trying to go because it's more clear information about the volatiles. Yeah. Sometimes they need a blend to be as a signal, but sometimes it's a specific one. For example, the citrus experiment, for example, was just the limonene. The nematodes was able to recognize and go for it. So
in the chestnut weevil example that you're working on now, you're just using nematodes that are naturally occurring in the environment, or are you able to spray nematodes on them and use particular nematodes?
So both. The way that I like to do those experiments, before I start anything on the soil, I collect a bunch of soil samples and trying to collect native nematodes. And if we can find some native nematodes, we try to multiply those and reapply, just increase the population, because we know for sure those nematodes are going to be happy on that environment. But sometimes we can't find, especially with conventional crops, when we have a bunch of pesticides going on, it's very hard to find native nematodes on the area. And when those things happen, we go with the more general nematode species. Now we have more than 20 different species in the lab now, so it's easy for us to test which one is going to be the best one for each case. So, and we decide, because the nematodes, they stay in different layers on the soil. And normally we choose a combination with some that goes more deep in the soil, the other ones that stay more on the surface, and apply those as a biological control. Yeah. And
I guess one more question. So when you look at the nematodes that are attracted to these volatiles, are they all pathogenic, or are there some that are non-pathogenic and are still attracted? So yes, in some cases we do work with plant parasitic nematodes, because this sign, it's easy for entomopathogenic nematodes to recognize and go through. How about the plant parasitic nematodes? That is going to be a big problem for the plants. Yeah. So I have some studies that prove that some volatiles could be attractive for both. And other cases is just for one or another one. The best way depends what crop you are working, test both. Okay. Thank you.
Well, Camilla, thank you very much.
Thank you.
It was great to have you, and glad to have you at UNC Asheville, and great to hear about your research.
Thank you very much. Thank you for the opportunity.